Hello everyone, so we are going to start the part 2 of amino acid metabolism. So here we are going to study about, first of all, the catabolism of amino acid. So what are the different processes by which the amino acid it get catabolized in our human body? So basically, if you see the catabolism of the amino acid, it occurs in the following way. First of all, it may undergo transamination. Moreover, it can undergo oxidative deamination. Moreover, it can undergo ammonia transport, and finally this will enter the urea cycle. So that is the overall, you can say, overall cycle of the catabolism of amino acid in the human body. So let's start with the transamination. So what is transamination? The word itself, it represents trans means the transport, amination means the amine group, that is the transport of the amino group. So what actually happens in this process? Basically, the transport of an amino acid group, it takes place from one amino acid to a keto acid, which led to the formation of a new amino acid and a new keto acid. So you can see in this figure, I am showing you amino acid alanine. So now when it combines with the alpha ketoglutarate, the resultant product, it is the pyruvate and the glutamate, and an enzyme which is responsible for this reaction, it is the aniline aminotransferase. Now you should know that it is catalyzed by a group of enzymes. This group of enzymes, these are known as the aminotransferases. This group of enzymes which are responsible for the transamination, these are known as the aminotransferases. And the cofactor for this transamination reaction, it is nothing but it is PLP, which stands for pyridoxal phosphate. This is the PLP, which acts as a cofactor for this transamination reaction. Now where it occurs? It occurs basically in the liver, kidney, heart and the brain, as these all organs, they have adequate amount of the enzymes which are necessary for the reaction of transamination. So now going to the next slide. Now what are the features of transamination? As this transamination reaction, it deals with the transport of the amino acid. So what are the features of this reaction? First of all the transaminases, all the enzymes which are involved in the transamination, they require the PLP as a cofactor, right? The next one is no free amino, means ammonia, no free ammonia liberated in this process of transamination. Rather, only the transfer of amino group will take place in the process of transamination. This transamination process, it is reversible in nature, so it can take place both by the forward reaction as well as by the backward reaction. Moreover, there are multiple transaminase enzymes which vary in the substrate specificity. So for different substrate involved in the transamination reaction, you will have different set of enzymes. And the AST and ALT. Now what is AST? AST is nothing but the aspartate aminotransferase, and ALT is the alanine aminotransferase. They make a significant contribution for the process of the transamination. And this transamination, it is important for redistribution of the amino acid groups, so which is involved in the production of non-essential amino acids in your body. Now you should know that it diverts, this transamination reaction, it actually diverts the excess of the amino acid. Whatever is produced in your body, it will divert this excess of the amino acid to the process of energy generation, or you can say this amino acid will be utilized for the synthesis of the ATP in the human body. Moreover, the amino acid which undergo actually the transamination, they finally concentrate the nitrogen in the glutamate. So the final acceptor of the nitrogen, it is the glutamate, and which involves the reactions of the transamination. Now this glutamate, now once this nitrogen group from the amino acid is actually get adopted, or you can say it is this actually get transported to the glutamate. Now this glutamate will undergo oxidative deamination. This glutamate will undergo oxidative deamination to liberate the free ammonia. It's the glutamate only which will liberate the free ammonia for the urea synthesis in your body. Now all the amino acid except the few like lysine, threonine, proline and hydroxyproline, they participate in the transamination reaction. But this enzyme, this amino acid like your lysine, threonine, proline and hydroxyproline, they don't participate in the transamination reaction, and it involves this transamination reaction is involves both the anabolism as well as catabolism. Thus this transamination reaction, it is regarded as a reversible reaction. Now let's have a look. So here you can see this is the amino acid 1. Suppose 1 I have designated as, just for example, this is amino acid 1. It will act with the alpha ketoglutarate to form first, that is the keto acid 1. Along with that you will have the glutamate, right? Now this alanine, it will, for alanine it will combine with the, if the alanine is the amino acid, it will combine with the alpha ketoglutarate to form the pyruvate and the glutamate. Similarly, if the amino acid it is the aspartate, it will combine with the alpha ketoglutarate to form the oxaloacetate and glutamate. So this is the reaction by which the transamination is takes place in the body. So here you can see I have given you one example, the alpha ketoglutarate, and this is the amino acid. So you will finally get a glutamate and a alpha keto acid. So this is the reaction which generally takes place in the body. So here I am showing you the cellular protein, how it involves the transamination, how it enters the liver, what are the different processes and the conversion of this amino acid. It is shown here. Now the mechanism of transamination. So what are the mechanism of transamination? Basically you should know that the transamination reaction, it takes place in the two basic steps. The first steps, that is the step 1, it involves nothing but it involves the transfer of amino acid from the amino acid AA1, what I have shown you earlier in the slide. So AA1, it is nothing but we have designated a number 1. So AA stands for the amino acid. The first step, it is the transfer of amino group from the amino acid to the coenzyme PLP, and this will form, once the amino group it is transferred to the PLP, it will form the pyridoxamine phosphate. This is the step 1, right? Now the amino acid 1, it is converted to keto acid 2. So this 1, 2 numbers are designated as per the state of the reaction they are appearing in the transamination process, right? Now what is the step 2? So step 2 is nothing but it is the steps where the amino group of the pyridoxamine phosphate, what it has formed in earlier, this is the pyridoxamine phosphate. Now this pyridoxamine phosphate, it is then transferred to a keto acid 1, which has already formed in the earlier step, keto acid 1, to produce a new amino acid 2. That is the sequence after the transfer. Amino acid 2, it is a sequence that forms after the transfer, and the enzyme with the PLP, it is regenerated. This PLP will be regenerated for the next transamination reaction. So now what is its significance? What is the significance of transamination? So basically, first significance is that the enzyme, it is present within the cell, right? It is now this enzyme, it is present within the cells. Now it release in the cellular damage into the blood. So if the cell it get disrupted, or if the cell it get damaged, then these enzymes which are responsible for the transamination reactions, they get distributed into the bloodstream, right? Now increase of the AST, what I have already told you, AST stands for the aspartate amino transferase. Now increase of the AST in the blood, it can lead to the myocardial infarction, right? Moreover, increase of the AST as well as ALT. ALT is the alanine amino transferase. It can cause the hepatitis, even it can cause the alcoholic cirrhosis, and moreover it can cause the muscular dystrophy. Now the next part, it is the transdeamination. So here what actually happens, the amino group of most of the amino acid, the amino group of the most of the amino acid, it is released by a coupled reaction. This reaction, it is known as the transdeamination. For example, if somebody asks you define transdeamination, it is nothing but it is a simple process where the amino group of the amino acid, it is released by a coupled reaction, right? Now this transamination, it is always followed, means the transamination, the next step of the transamination, it is the oxidative deamination. So the transamination, it is always followed by a oxidative deamination. Now moreover, the transamination, it takes place in the cytoplasm of the cell, right? And the amino group which is transferred, actually it is transported to the liver as a glutamic acid, right? And which is finally oxidatively deaminated in the mitochondria of the hepatocytes. So this is the process by which the transdeamination takes place. So this amino acid will be first of all it will be transported, then it will form the glutamic acid. Now this glutamic acid will be oxidatively deaminated in the mitochondria, right? Now deamination. So what is deamination? D means to remove, means something which is removed, and amination. Now amination means the removal of the amino group. So the removal of the amino group from the amino acid as ammonia, it is known as the deamination. This is the basic definition, right? Now the deamination results in the liberation of the ammonia for urea synthesis. Afterwards, after the process of deamination, the ammonia will be released and it will be used for synthesis of the urea in the human body, and the carbon skeleton of the amino acid is converted to the keto acid. So the carbon skeleton, so whatever the skeleton, suppose for aromatic amino acid you will have a aromatic carbon skeleton, for a aliphatic amino acid you will have a aliphatic carbon skeleton. Now this carbon skeleton is generally get converted into keto acid, right? Now the deamination may be either oxidative or non-oxidative. That is a sequence, means as per the convenience it will be either the deamination process will be either oxidative deamination or it will favor the deoxidative deamination. Moreover, what actually the thing that to be keep in your mind in this process of the deamination is that only the liver mitochondria contain the enzyme which is known as GDase. GDase, it is nothing but the glutamate dehydrogenase. So only liver mitochondria have the glutamate dehydrogenase, which deaminates the glutamate to alpha ketoglutarate and ammonia. So GDH, it is an important part for the process of deamination. It's an important part, you can say it's an important enzyme for the process of deamination. Now it needs this enzyme, it needs, or the process, it needs NAD positive as the coenzyme for this reaction. Moreover, this enzyme, it is nothing but it is a allosteric enzyme. So it is activated by the ATP and inhibited by the GTP. So it have two sides. So one is the initiator side and one is the inhibitor side, right? Now oxidative deamination. So in the oxidative deamination, you should know that oxidative deamination, it is the liberation of free ammonia. Here the free ammonia will be liberated from the amino acid, right, coupled with oxidation. Now this ammonia will be utilized, as I already told you, this ammonia will be utilized for the synthesis of the urea, right? Now where is the site? The most common site, it is the liver and the kidney, but preferentially it takes place in the liver. But as I already told you, this enzyme, particular enzyme, that is the glutamate dehydrogenase, it is needed for this process, right? And this oxidative deamination is to provide the ammonia for urea synthesis, which I have already told you, and alpha ketoacids for variety of reaction, which includes the energy generation. Now from where the alpha ketoacid will come? The alpha ketoacid will come from the chain. After removal of the amino group, the remaining chain, it will get converted into the ketoacids, right? Now what is the role of glutamate dehydrogenase? We are speaking about the glutamate dehydrogenase as it is taking a vital role in the process of deamination. So what is the role of glutamate dehydrogenase? So first of all, you should know that this glutamate, it is recognized as a collection center for amino groups. This glutamate, it is recognized as a collection center, means it will collect all the amino group from the amino acids, right? Now this glutamate, it rapidly undergo oxidative deamination. That is its fast property. The process of deamination is quite fast with the glutamate dehydrogenase. Moreover, it is catalyzed, this reaction is catalyzed by the GDase. The deamination was catalyzed by the GDase, as we have already discussed, to liberate this ammonia, and this ammonia will be utilized for the urea synthesis, right? Now it will use either NAD positive or NADP positive as a cofactor for this reaction, and this conversion occurs through the formation of alpha aminoglutarate. I'll show you what is the reaction, how it takes place. So basically, see here, this is the L-glutamate, right? This is the L-glutamate. Now here GDH will occur. Now this is the cofactor NAD positive. It is the cofactor, right? Now you can see here it has formed the alpha aminoglutarate, right? And you see here this NH3 positive amine group, actually it has now reduced to amino group, means NH group, right? Now from here you can see finally you will get a alpha ketoglutarate, or it will finally get converted into alpha keto acids, and this ammonia is finally released. This ammonia is released, and this will enter into the synthesis of the urea. Now metabolic significance. What is the metabolic significance of this reaction? First of all, this reaction is a reversible reaction, so you can say that both the anabolic as well as the catabolic process, it takes place in this reaction. Moreover, what is going to regulate the GDH activity? So basically GDH activity, it is regulated by, basically the regulation is, it's a zinc containing mitochondrial allosteric enzyme. You should know that, right? It consists of this GDH, it consists of the 6 identical subunit, right? Moreover, the molecular weight, it is 56,000. So this is the GDH, and these are its property. On based on this property, anybody or any substance can, or any substrate can control the activity of GDH. Now allosteric regulation. So how allosteric regulation takes place? So GTP and ATP, these are the allosteric inhibitor. I will give you in details how it takes place. GTP and ATP, these are the allosteric inhibitors for the GDH, right? Moreover, GDP and ADP, these are the allosteric activator, right? Now you should know that the GTP and ATP, they will inhibit the process, whereas the GDP and ADP, they will activate the process, right? Now less energy in the body, or less amount of the ATP, will result in the increase in the oxidation of the amino acid to meet the energy demand of the body. If the energy is less, if the ATP is less, then the oxidation of the amino acid will increase, the rate of oxidation will increase. Now steroid and thyroid hormones, you should know that it inhibits the GDH activity in the human body. So here you can see this is the glutamate. It has converted into alpha ketoglutarate, and this amino group, it has come out from here. Now here you can see the GTP. So this GTP, it is a allosteric inhibitor, and whereas this here I can show you the ADP, this is a allosteric activator for this reaction. So this is actually a reversible reaction, right? Now amino acid oxidases, another set of enzymes which are responsible for the metabolism of the amino acids. So L-amino acid oxidase and D-amino acid oxidase, these are the two important enzymes, L-amino acid oxidase and D-amino acid oxidase, right? Now flavoprotein and the cofactors are for this enzyme, the cofactors are the FMN and FAD. Now where it will act, these enzymes, they act on the corresponding amino acid to produce the alpha keto acid and ammonia, right? These amino acid oxidases, they will act on the amino acids to produce alpha keto acids and ammonia. Now where is the site? It is the liver, kidney and peroxisome. So these are the site for the production. Moreover, you should know that the activity of L-amino acid oxidase is very low. The rate of activity for L-amino acid oxidase is very low, and this L-amino acid oxidase, actually it has a minor role in amino acid catabolism. Now this L-amino acid oxidase, they act on all the amino acid. You should know that this L-amino acid oxidase, it acts on all the amino acid except the glycine and the dicarboxylic acid. As we all know that this is the glycine, it is the simplest amino acid. So L-amino acid oxidase, it don't have any sort of activities on the glycine, right? Moreover, the activity of the D-amino oxidase is high than that of the L-amino acid oxidase. So therefore you see here, I have told you earlier that amino acid oxidase, it have two subtypes. One, it is the L-amino acid oxidase, and one, it is the D-amino oxidase. Now the activity of this D-amino oxidase, it is always high as compared to the L-amino acid oxidase. Now this D-amino acid oxidase, it degrades the D-amino acid in the bacterial cell. So D-amino acid oxidase, it have a role in the prokaryotic cell, thus it degrades the D-amino acid in the bacterial cell wall. So oxidative deamination of the amino acid. So diagrammatically it can be shown as, suppose this is the L amino acid, right? Here L amino acid oxidase will act to get a alpha keto acid and an ammonia group. So here the cofactor are FMN, as I already told you, the cofactor it is FMN, right? And finally it led to the liberation of a peroxide, a water molecule. Now the fate of D-amino acid. What will be the fate of the D-amino acid in this reaction? So first of all, you see this D-amino acid reaction. So first of all, this FAD will act, right? Here, apart from that, there is a hydrogen water molecule is involved in this reaction, right? It will form the alpha keto acids, and this alpha keto acid may further get converted into L-amino acid and glucose along with fat, and this glucose can be utilized for the production of the energy. So basically the D-amino acids are found in the plants and microorganisms. That is the first thing. Moreover, they are present in the mammalian proteins. This D-amino acid, they are present in the mammalian proteins, right? And the D-amino acids, they are taken in the diet or bacterial cell walls, absorbed from the gut in the human body. If the D-amino acid is present in your diet, it will be absorbed from the gut. And this D-amino acid, this oxidase, amino acid oxidase, this enzyme particularly, it converts the D-amino acid to the respective keto acids. So here you can see how it gets converted. This D-amino acid, it gets converted into the keto acid, right? This alpha keto acid, it can undergo transamination. Here you can see it undergo transamination to be get converted into L-amino acid. Here you can see how it has get converted into L-amino acids, and it participates in the various metabolic pathways. And this keto acid may be oxidized to generate. Here you have seen, here it is oxidized to generate the glucose and the fat, and this glucose can be utilized for the genesis, or you can say the genesis of the energy. Now, what is the non-oxidative deamination? That the earlier one, that is a oxidative deamination. The next one, it is a non-oxidative deamination. So this is the example I can show you. So very basic thing is that the direct deamination without oxidation. Non-oxidative deamination means it's a direct deamination without oxidation. Here oxygen has nothing to play. Now amino acid dehydrogenase. This is the enzyme which is responsible for the process of non-oxidative deamination. This is the amino acid dehydrogenase, right? So we are taking here example of 3 amino acids: serine, threonine, and homoserine. These are the hydroxyl amino acid. Now when on this amino acid, the dehydrogenase, it acts, right, you will find a liberation of ammonia along with the formation of respective keto acids. Now amino acid desulfidase, this is another set of enzyme. How it acts. So you can see where I have taken an example of an amino acid which is known as the cysteine. Now on the cysteine, if the desulfidase it acts, it forms the pyruvate along with the liberation of hydrogen sulfide and the ammonia. So the cysteine and the homocysteine, they undergo the deamination coupled with desulfidation to give the keto acids, right? One more example I have given you here, that is the deamination of histidine. What will happen when deamination of histidine will take place? You can see here the histidine, when deamination of the histidine takes place in the presence of the enzyme histidase, it will form the uroconate, and a ammonia molecule will be liberated. This will further enter into the urea or urea cycle, right? So thank you everyone. I am going quite fast, but hope this presentation will enlighten you regarding the process of the deamination and transamination which takes place in your body on the amino acids. So if you like the video, you can press the like icon. You can share this video with your classmates and colleagues, and finally, for further notification from this channel, you can press the bell icon. Thank you.